This is Dr. Terry Stull, and I'm going to present bacterial vaccine polypeptides, a novel method that will add a new tool for a new class of bacterial vaccines. So there's a great need for bacterial vaccines, not only an unmet need, but also opportunities to address some of the limitations of even the current childhood vaccines. Unfortunately, we don't really have the tools to address those right now. However, in the world of virology, you know the impact of structural vaccinology, especially on the recent epidemic vaccines. But there's not really good structural vaccinology tools in bacteria, and so we intend to address that gap. I'm going to talk about non-typable Haemophilus influenza because it's such a common cause of infection in children. It's such a common cause of infection in adults, especially with COPD. And the current vaccine does not address any of the strains that cause these diseases. Although I'm going to talk about Haemophilus influenza, we have data that indicate that the methodology that I'm going to describe to you is useful in other bacteria as well. So if this is the inside of the bacteria and this is the outside, we're searching for needles in a haystack. So we're looking for the regions of proteins on the surface that are really the very best vaccine targets. And when we find those regions, we figuratively pick them up and fuse them. Of course, this polypeptide eventually is encoded by a simple plasmid, but that's the general approach. Now, how do we do this scientifically? Well, we use bioinformatics to begin with, and we're searching for core surface proteins. These are the proteins present in all the strains in the species. And then we subject those to a protein structural analysis, and then we go back and examine the sequences. And when we finish doing this, we're searching for the surface exposed, sequence conserved regions. In the next step, we use another criteria. In this case, either the individual peptide is protective in an animal model, or more recently, we've used quantitative expression, which is also very useful. Finally, we take these polypeptides constructed as I described for you and test with routine traditional immunogenicity and protection models. So chinchillas are the gold standard model for ear infections. And in this case, we injected chinchillas with our polysaccharide or our polypeptide for Haemophilus influenza. And you can see that it behaves like a routine protein with an ELISA titer of 1 to 135,000. This log2 graph demonstrates that every single peptide was immunogenic, and as you would expect, some more than others. There are multiple measurements, and this is just one representative measurement of the intensity of ear infections. And here you can see that the vaccine group was clearly protected compared to the unimmunized group. And similarly, as we began to look at the potential of COPD, addressing COPD, we found with a mouse clearance model that the immunized group was also protected compared to the unimmunized group. So bacterial vaccine polypeptides are an innovative approach that is multi-targeted. Every target is present in every strain of the species. Every target is 100% sequence conserved. The targets are surface exposed for immune accessibility. The construct of bacterial vaccine polypeptides is immunogenic, and it's effective in multiple preclinical models. And fortunately, it's also easily manufactured as an E. coli recombinant protein. Thank you.